In this video, we will take a closer look at enzymes. So remember, enzymes are a type of protein, and their job is to catalyze reactions. This means that enzymes are responsible for speeding up chemical reactions occurring inside of organisms. Every enzyme, because it's a protein, has several levels of structure. The tertiary or quaternary structure gives this enzyme complex shape with many nooks and crannies. One of those nooks is called the active site. This is the location on the enzyme where the chemical reaction occurs. The active site is very specifically shaped, and it matches only one type of reactant or substrate that the enzyme will work on. In the example here, we see the enzyme lactase, which is responsible for breaking down milk sugar, lactose. The active site is this groove where the lactose sugar will nestle into the enzyme. This is where the chemical reaction is going to occur, and the bond between the two sugars in lactose will be broken. Again, the substrate is the molecule or molecules that the enzyme works on. These are the reactants in the chemical reaction. Every chemical reaction requires activation energy. This is the amount of energy needed to start the chemical reaction. Enzymes work by lowering the activation energy of a chemical reaction. We can look at the activation energy on a graph. So on one side we have the amount of energy required. On the other side of our graph we have the progress of the reaction. Every chemical reaction will start with our reactants. The activation energy is this bump that must be overcome before the reaction can really get going. And then we have the reaction go to produce our products. Enzymes work by lowering the activation energy. Nothing else about the reaction will be changed. We have the same reactants and the same products. The only difference is this hump in between. The enzyme will lower the activation energy, leading us to the same products. This speeds up the reaction overall. We can think of the activation energy as a barrier that must be overcome. So in this example, we have the reactants, the dry frogs, and they have to overcome this rock wall barrier to get to the water and become the products, wet frogs. Without enzymes, we see that this is a very steep barrier that they must overcome. Okay. When we add enzymes to the reaction, this is going to lower the energetic barrier and make it easier to get over the hump and into the product zone. Okay. So this helps us speed up chemical reactions and makes them run much more rapidly inside our bodies. There are many ways that enzymes reduce the activation energy. The first way is by stressing, bending, or stretching critical chemical bonds. So in our example of lactase, the lactase takes the bond between the two sugars and stretches it so that it is strained and easier to break. Another way to lower the activation energy is by directly participating in the reaction. Some enzymes get directly involved in everything. They grab hold of the substrate, participate in the reaction, hold those transitional molecules before producing the products. A third way to lower activation energy is by creating a microhabitat that is conducive to the reaction. So in some cases we have molecules that are hydrophobic. An enzyme might take those hydrophobic molecules and wrap them up in a hydrophobic environment. This allows the reaction to occur more rapidly. Remember, cells are full of water, so if the molecule is hydrophobic, it's not going to want to be in that water. The fourth way that we can lower the activation energy is by simply orienting or holding substrate molecules in place so that they can be modified. So enzymes are good at grabbing hold of molecules and putting them in the correct orientation or the correct location so that they can react together. Without that enzyme, those molecules bouncing around randomly would have to bounce together in the correct orientation all on their own. Enzymes are sensitive to the conditions of the environment around them. This can include the concentration of chemicals around them, as well as things like temperature and pH. We're going to look at several physical factors that affect the rate of reaction of an enzyme. The first factor we're going to investigate is the enzyme concentration. So, as we add more enzyme to the solution, we're going to see the rate of reaction increase. So this is a fairly linear trend. We can also look at the substrate concentration. So how much reactant is there for the enzyme to work with? As we add more substrate, we're going to see that the rate of reaction will increase for a time, but eventually it levels off at a maximum rate. This peak or maximum rate occurs because sometimes all the enzymes are busy. At that point, the reaction cannot go any faster. Another factor we can look at is temperature. Temperature is very important for proteins in general. If you heat up a protein too much, it's going to lose its shape and denature. On the opposite end of the spectrum, enzymes that are too cold do not react very rapidly either. This is because when we measure temperature, we're measuring molecular movement. So if we are very cold, that means the molecules are not moving very much, which means they're not interacting with each other at a very rapid rate. This slows the overall reaction rate of our chemical reaction. So if we look at our temperature and we start at 0 degrees Celsius and we go to say 100 degrees Celsius, okay, every enzyme has an optimal range. We're going to see that the reactions are very slow when it's cold. They increase as we reach that optimal point. And if we get too hot, the reaction rate is going to fall back down to 0 as that enzyme is denatured. So every enzyme has an optimal temperature where their reaction rate is at its maximum. For many enzymes found inside our bodies, the optimal temperature is near body temperature, which is 37 degrees Celsius. Enzymes also have an optimal pH range, and enzymes are very sensitive to pH changes. We can see that the pH scale goes from approximately 0 to 14, okay, with 7 being neutral. Most enzymes inside of living organisms have an optimum pH of around neutral, or 7. Okay. At either extreme, either extremely acidic, near 0, or extremely basic, around 14, enzymes will denature and lose their shape. Though many enzymes have an optimal pH of around 7, that is not true of all enzymes. One good example is the enzyme pepsin, which is found in your stomach. Remember, your stomach has a pH of around 2. If the optimal pH for pepsin was at 7, the enzyme would be denatured in those conditions. Instead, pepsin has an optimal pH range of around 2. So it peaks around 2 and is denatured around neutral conditions. So pepsin is an exception to the rule of 7 or neutral being an optimum pH. Cells often regulate enzymes and can turn them on and off. One of the most common ways that cells regulate their metabolic pathways is through binding of other chemicals to the enzymes. This can alter the enzyme shape in a way that increases or decreases the enzyme's activity. Some chemicals act as an activator. Instead of binding to an enzyme and turning it off, their binding to the enzyme turns it on, altering the enzyme's shape or structure so that it can now catalyze a reaction. So we can look at the rate of reaction with the amount of activator. The more activator you add, the faster the rate of reaction is going to be. To turn enzymes off, we can use inhibitors. These reduce enzyme activity and come in two types. There are competitive inhibitors, which bind to the active site itself, blocking the substrate molecules from entering that site. And there are non-competitive inhibitors. These inhibitors bind to a different site on the enzyme and change the shape of the active site, preventing the substrate from binding. Both of these slow the rate of reaction. So the more inhibitor we have, the slower our reaction will be. Enzymes are important in our bodies because they are participating in most of the chemical reactions occurring inside of us. One big one that we're going to talk about in Chapter 4 is cellular respiration. Enzymes are responsible for keeping this reaction running so that we can produce ATP energy that actually powers our cells. Plants also rely on enzymes for photosynthesis. This is another major energetic process that harnesses solar energy from the sun and transfers it or converts it into chemical energy and sugars. Without enzymes, these major reactions inside of our body would not happen at an efficient rate. Enzymes are important in our metabolism. When we talk about metabolism in biology, we're discussing all the chemical reactions in your body. This means making and breaking molecules. So we can make big molecules like those proteins and carbohydrates. This is part of digesting food, making new components for the cell, and energy processing like cellular respiration. This video has covered enzymes. Remember, they are proteins that catalyze reactions. We've looked at how external factors can affect enzymes and how extreme conditions often denature those proteins and make them lose their shape. Enzymes are important because they're used in pretty much all the chemical reactions of your body. In the next chapter, we're going to investigate some of the major energy processes that occur in nature, photosynthesis and cellular respiration.